All right, good afternoon, everybody. Welcome to week 9. I have no idea how we got to week 9 so quickly, but here we are. So um, today, today we'll be finishing up our lecture on population ecology, the two-parter from last week. We have a few slides left, and then we'll be moving on to microbe-microbe interactions. All right, we still have a few students coming in. So we left off talking about, if you remember, we were talking about concepts like ecological successions. Can somebody give us a real brief definition of what a community succession was from last time? Who remembers what an ecological succession was?
Is it like there are four different ways, and one of them was like preparing for other species to inhabitate, like the facilitative autogenic succession?
Right. Yeah, I guess I was meaning just in general. What do we think of? There's different types of them, but in general, what is a succession? Like what
Behavior,
happens?
behavioring of different species after one another, like
Okay.
continuous growth and death of species communities.
Exactly. And so there's different kind of flavors or ways that these appear in nature. Um, and the concept of a climax community is almost the end of that process in a sense. So the climax community represents an equilibrium state where at the end of a series of successions in your community, you end up settling on a stable community. So it's not constantly changing all the time. So this is kind of a theoretical construct because in the real world, it's hard to identify these, particularly because we've talked about some of these things that can cause successions like seasonality or changes in the environment, a forest fire or disturbance of some sort. Many of these things would reset the clock on these successions. And so identifying kind of an end point or a so-called climax community can be challenging. Probably one of the best systems for this would be a rumen. So ruminant animals like cows, and we're going to talk about this in a few lectures, have a very complex symbiotic association of all three domains of microbes. And in that rumen, on a certain type of food, right, like cow feed, grass, and other, sort of grains that they're fed, you can end up with a stable community. And that's actually what farmers want to see. And we'll talk when we have the lecture on rumen microbiology about some of the things that can disturb that. So this equilibrium would bring about homeostasis or stability, right? It doesn't mean the community doesn't change at all, but it needs to be predictable. So with seasonality, you can actually have a climax community that changes over the seasons. But if you end up with the same communities recurring year after year after year, then you could also consider that to be kind of a terminal climax community assemblage. So for example, phytoplankton blooms that sequentially happen, as we've talked about several times, might be still kind of considered a climax community. But in general, we're talking about communities that don't really change a whole lot. And so this is not the norm because disequilibrium really seems to be the norm, right? And this actually leads to enhanced diversity. And there was a famous macroecologist, Joseph Connell, who identified what he called the intermediate disturbance hypothesis in studying intertidal organisms like invertebrates in uh, up in the San Juan Islands in Washington State. And so what he observed was that periodic disturbances, like, say, a log coming into the intertidal and bashing a bunch of rocks and organisms and clearing the rocks away, um, or predation occurring through, like, he was one of the ones who identified keystone species of predators like a sea star in the intertidal. So he actually said that a little bit of disturbance is good for diversity because it creates patchiness and variability, right? And so what this does is it resets the successional sequence, as I mentioned a couple of minutes ago. These kinds of disturbances can actually enhance diversity because you don't end up with a stable equilibrium climax community. Instead, you end up with different parts of a habitat being at different stages in the successional sequence. And so we end up with a greater overall diversity if we consider that entire ecosystem. Does that make sense? Now, there was another thinker that worked independently. So G. Evelyn Hutchinson, we've encountered them before because they were the ones that came up with the n-dimensional hypervolume concept for niches. But Hutchinson identified what he called the paradox of the plankton. And this was also kind of discovering that diversity seemed to be higher than you might otherwise predict. If we focus on phytoplankton, if I said, tell me what phytoplankton do, what would you respond? What do phytoplankton do?
Produce?
Okay, so they're primary producers. Is that what you mean?
Yes, primary producers.
Okay, so they're photosynthetic. They're primary producers. Um, what do they eat? So-called eat, or what do they really need? What do algae really thrive on? Let's see. Somebody put something in the chat. Let's open that up. Jose says light. I'll buy that. What else do they need? If you're going to be phototrophic, you need light. What kind of nutrients do you especially need?
Carbon dioxide.
Okay, CO2, right? Which is pretty plentiful, right? Because it's in the atmosphere and it dissolves in water pretty easily. What else? What have we talked about in terms of, remember, eutrophication? What does that involve? How quickly we forget. Eutrophication involves what? Nitrogen. Okay, thank you, Jennifer. Yeah, so nitrogen and phosphorus, right? Those key essential nutrients. Thank you, Angel. So we talked about competitive exclusion last time, right? And so in theory, all phytoplankton should be competing with each other, right? Because they all need light, they all need CO2, and they all need nitrogen and phosphorus. And yet what Hutchinson found is that if he went to, a local beach, a bay, or he went to a lake, you don't just find one species, you find multiple species of phytoplankton. And in fact, a more recent study in San Francisco Bay identified over 500 species of diatoms, just diatoms, right? So these are the silica-based phytoplankton that would need all of these things plus silicon. And yet there's so much diversity there. How is that, right? They should be out competing each other. But essentially what he hypothesized is that there's lots of variability in the environment. So those day-night cycles, those seasonal cycles, those um, differences in parts of San Francisco Bay from other parts, other biological interactions like predation that Connell mentioned up above, all of these things together combine to create the opportunity for different species to coexist without driving each other to local extinction through competition. So it reduces the impact of competitive exclusion. And so this was what he described as the paradox of the plankton. Okay, everybody with me on that? So even though they're all using the same nutrients, the same light, the same CO2, they're not competing for it on level playing fields because of other environmental factors that come into play. Okay. And so that's how you can end up with lots of different diversity of phytoplankton coming from a single scoop of seawater, for example. Okay. And so ultimately, we see these limitations of competitive exclusion and changes in competitive status. All right. Any questions about that? Okay. So these two things are driving us towards increased diversity, whereas the climax community would actually lead to an equilibrium state with lower diversity. All right. So, excuse me. All right. So the last little bit of the lecture is focused on how we measure diversity. So I keep using this term, right? Okay, diversity of phytoplankton, diversity of this, diversity of bacteria in an environment. But what do I really mean by this? We actually use this term incorrectly in many cases. I think we all equate diversity with one thing in our brain. So if I said, what is the diversity of bacteria in a sample of seawater? What does that bring to your mind? What do you think that means? If I asked you to figure out what the diversity of, what would you count?
Different species?
Different species. Exactly, Carlos. So it turns out that that's not diversity. So from a strict definition standpoint, that is richness. Okay. So there's this term, species richness. And the way this is calculated, excuse me, sorry, this keeps going haywire. So what this does is it takes the number of species, S, and we subtract one. That's a mathematical thing, right? In stats, you learned you have to take n minus one for a lot of calculations. You don't need to worry too much about it. But basically, we're taking the number of species and we're putting it over the log of the number of individuals. Okay. So let's say you have 10 species of diatoms in a sample, right? And you have 100 individuals, right? So you counted 100 cells and you identified them by species, and they might be equally distributed as 10 and 10 and 10 and 10 or differently, right? But you end up with 10 minus 1 over the log of 100, which is 10. And so in that case, richness is going to be pretty near to 1. Okay. What that value of richness does is it takes into account how many species you have, but what is it missing? What doesn't it tell us? And if you look down here at this Shannon Weaver diversity, maybe you can kind of get a hint as to what the limitation of the richness metric is.
The abundancy?
Right. So the relative abundance, right? Okay. So, and that's also what Jose put in the chat. So if we have 10 species and 100 individuals, it could be 10, 10, 10, 10, 10, 10, 10, right? Or it could be 91 and 1, 1, 1, 1, 1, 1, 1, 1, 1, right? Or anything in between. And obviously, those two communities would be very different. And what that does is that gives you a difference in what's called evenness. So evenness of a community is a measure of how evenly distributed the individuals are across the species in a sample. So if you had 10, 10, 10, 10, 10 all the way to 100, that would be a perfectly evenly distributed community. Do you think we see that in the real world very often? Do you think perfect evenness is common?
No.
No, it's not.
Why not? You're absolutely right, but why wouldn't that be the case? Do you think? Any ideas?
The biotic factors.
Okay.
Yeah.
So maybe some are a little better at tolerating conditions or thriving under a certain set of conditions. Okay. Um, Essentially, we talked a lot about competition, right? Are all species equally good at competing? They're not, right? So we typically expect there to be uneven distributions. And so what we do is we calculate the Shannon-Weaver index of diversity. And what this does is it accounts for the number of species, but also P here is the relative proportion of individuals in each species. And you don't need to worry why, but for mathematical purposes, we take P and we multiply it by natural log of P. But basically what you're doing, what I want you to take from this equation is that we are summing, right? We're summing the relative proportion of individuals in species 1 and species 2, species 3, so on and so forth, until you end up with the ith number of species, right? However many that is, right? In the case I gave you of the example, it's 10 species. And so if you end up with even distribution, then your relative proportion will be the same for each one. If you end up with a bias towards, one or two really abundant species, and then the others are more minor players, that will change the values that come out of this. And because we're taking P log natural log P, we're going to end up with a negative number. And so you put a negative in front of it all to make it positive. Okay. So let's just look at an example. On the final, I'm not going to ask you to do this calculation. I just want us to understand the concepts and what the difference between richness and diversity is. Okay. And these are not the only ways to calculate this, but for our class, they will be. So population 1 on the left, what do you notice about that population? Like, what's distinct about that one on the left? So for the one in blue, for example, how even is that one? Jose puts in the chat that it's even proportions. So this is the example like I was giving a second ago. Instead of having 10, we have 5. So they're each 20% instead of 10%. But same difference, right? So the one on the left is a perfectly even population, and that's reflected in the E being 1. Okay. And so it turns out if you take the diversity index and you divide by the number of species, then that tells you how even it is. Okay, the closer it gets to 1, the more even. Okay. But let's take a look at this. So S, we said, was just the number of species. Population 1 and population 2, how do they compare for S? So for the blue and the yellow, how do they compare for the S? They have the same. And so what does that mean? Johan, I saw you put a question mark after the same, but what does it mean to have equal S? I just want to make sure we all understand what this means.
Would it be the same amount of species being compared?
It is. So if I go to an environment and I sample it, I have five species in sample one, take another sample, 5 species in that one. That doesn't mean these are the same exact communities though, right? Because if you look at some of the other values in these two columns or sets of columns, we see some differences, right? So if all we looked at was the number of species, which remember is what we often associate with diversity, it's not really gonna give us a full picture. In fact, when we calculate richness, remember that was D up here. What do you notice about the population 1 and population 2? How do they compare for D? Yeah, they're also the same. Thanks, Ashlyn, right? And the reason they're the same is D only has two variables in it, S, which we said was identical, and N. And in this case, we counted like 100 individuals or whatever from two populations. So N is identical. Okay. So it's only if you calculate Shannon-Weaver diversity, H, that you decide or you determine that there's a difference in the community. And if we had looked at our relative proportions, you knew that all along, right? Because here, the first two species make up 75% and the remaining 3 are only 25. Okay. So there's a bias in the community distribution here, which is normal, right? And what you can see is that the value for H goes down. And so it turns out that diversity, actually, that metric is maximized when evenness. So the greater the evenness, the higher the Shannon diversity. It's also impacted by the number of species and things like that as well. But in general, you're going to end up with higher H for the same number of species. You get higher H if it's more even and lower H if it's less even. And so that's why this number is here. Now, this doesn't mean that this community is any less stable, any less healthy. It just means it's different. Okay. And so the Shannon Diversity Index allows us to determine and observe that difference. Does that make sense? All right. So the last thing I'll show you is, and I didn't put this in your slide, but this is just some work from some of our own studies, actually now quite an old study in my lab where we did some what are called rarefaction analyses. And this is a pretty common approach when you look at, for example, 16S ribosomal RNA sequences from an environment, as we've learned about, right? What you can do is you can ask the question, how many species do I have in my sample? But remember, for microbes, we said that species definitions can be tricky. So instead of using species and like interbreeding, what did we say we can use if we're studying 16S genes? What was the cutoff? Does anybody remember?
Is it 97% for 16S?
Exactly. So if we set our OTU or operational taxonomic unit threshold at 97%, everything that's 97% and above will be considered one species. And if it's below when we compare them, then we would call them two species. Okay. And so what I did, or my students did, is they counted the number of OTUs across different samples. These were some samples taken in the Salton Sea, which is southeast of here, out east of San Diego in the desert, kind of south of Coachella. And so what we found was that when we went sampling in November and August, we had lower numbers of OTUs, so lower species richness. This is not Shannon diversity, but the number of OTUs would be the equivalent of like S from before. And so what you can see from these rarefaction curves is that in April, in the springtime, when nutrient levels were high because of agricultural runoff, we ended up with higher numbers of OTUs, comparable with what we got in the sediments. And remember, we said that in sediments and soils, we tend to have higher numbers of species, higher diversity than you have in aquatic samples. And so that was just kind of something we saw. So this is another way that you can show the data. So what you do is you ask a question, you look at a sequence, and you could do this with a computer, of course, but if we did it manually, what you do is you take your sequence and you say, is this a new OTU or not? So I pick up my sample and I ask the question of the first one. Well, the first sequence has to be a new OTU because it's your first one, right? So that goes up. And so you get one new OTU for one new sequence sampled. But the height of this curve only goes up linearly if every time you grab a new sequence, it's a new species as well. And so what you can see is that in the spring, we had many more novel species. So as I grabbed new sequences, as we did 10 sequences and 20 and 40 and so on and so forth, we ended up with lots of new ones. Not 100%, right? At 80, we had 60 roughly. Okay. But in the other 2 months, at 80, we only had about 20 OTUs. So what that means is that we were grabbing duplicates when we sampled from our essentially clone library, right? And so rarefaction is a way for you to understand a couple of things. One is how many species are available, but also how well you have sampled your environment. And so in these 2 samples, it was leveling off. And so we felt like we had a lot of the species already in hand from this. But up here, we would have needed to keep sampling much longer before we had fully identified all of the so-called species or OTUs in that sample. So rarefaction curves help in that regard as well. So you're not required to know. I just like to share that because it's another way for ecologists, microbial ecologists in particular, to highlight diversity in an environment. Are there any questions about that last little bit? Okay, so we're
I do
moving
have a
on.
question.
Yeah, please do.
With diversity and richness, how, well, I'm trying to state this correctly. How accurate is it really in compared to, I guess, in a microbial environment when we're limited to what species we actually collect? Like, how do we count for error or the lack of collecting a certain species?
Yeah, well, that's a good question. So if you did a rarefaction curve, you'd have a sense of how kind of saturated you are. But of course, one way that we could figure out whether we have fully sampled things is we could also do replicates. Okay. So, and especially now that sequencing has gotten considerably cheaper, you'd want to go out and do multiple libraries. And of course, now we have next generation sequencing. That didn't exist when that study was done a long time ago. So nowadays, you would get thousands of OTUs because you'd be sequencing millions of 16S genes. So the technology has helped, but also you'd want to replicate your samples so that you could account for variability that might be, um, seen. But that said, we still have all those caveats. Remember, we learned about the caveats associated with PCR and the various methods. And so if you're going to sequence genes like 16S genes, it's never going to be perfect. Does that answer your question?
Yes, it does. Thank you.
Okay, cool. All right. So the next few lectures are going to be focused on kind of the interactions microbes have with each other and with other organisms. Okay. So microbes are, you know, integral to so much, okay? And of course, they don't just interact with us, but they interact with each other, and these can be classified. So what we're going to talk about over the next few lectures is really, okay, why is this? It says, you guys can't see that, can you? Ah, something went wrong with my PowerPoint. Hang on. Let's try this again. Let me try to share that. Okay, it says, can you see it? It says it's not showing.
Yeah, we can see it.
Oh, that's so weird. It says sharing is paused. Okay. Anyway, well, as long as you guys can see it, but let's see if it'll go forward. Okay. All right. Anyway, so these can be positive interactions, they can be negative interactions, or kind of neutral. Okay. And so we've already encountered some of this, right? So we've talked about intra-population interactions. What was one of the things we talked about in our last set of lectures within a population?
Sorry, doctor. It seems that it is paused.
Oh, it didn't
Yeah.
go forward?
No.
Ay, what's happening? Okay, so that's so weird. Yeah, because it's not showing up like a real PowerPoint. Let me try. Okay, let me. That is so strange. I wonder. This is not doing anything different than I ordinarily do. So I wonder if something happened when I copied over the. Let me see if I can share it without. So if I advance it this way. Okay, can you see that?
Yeah.
Yeah.
Okay, so we're just gonna have to go with this format, I think. I don't know why it's not letting me do the full screen, but this will work. And I'll try and figure it out before next time. All right, so it unfortunately means that probably the progressive disclosure is not gonna work for us though. So last time, or a couple of lectures ago, we talked about an intra or within population interaction. And what were we talking about there?
Competition.
Competition. We modeled that with the Lotka Volterra. And what did we say about competition? Is it good? Is it bad? What kind of interaction is it?
Always a bad one.
It's always a bad one, right? And so that's what's seen here, right? So growth rate is going to decrease for both species, or intra-population, it's just for that individuals of that one species, okay? But if you bring two together, then it's bad. So at high cell densities, even within a single species, you can end up with some negative interactions. But it turns out we can also have some positive, and on the next slide, we'll have an opportunity to kind of discuss that a little bit. Another possibility, and so these would be cooperative interactions, which are more common at low to moderate cell densities. In theory, you could have neutral interactions where you don't have a change in growth rate when interacting, but this would be pretty rare, especially for microbes, right? Because since we know that microbes like bacteria reproduce via binary fission, as soon as those two daughter cells form, they're competing with each other for resources, right? And they're side by side in physical proximity. So these neutral interactions are probably pretty rare, okay? So negatives are going to be very common, but let's talk about some of these positives. And so what I'd like to do is have a breakout. So it turns out that there's something called the Ali effect, where in some cases, larger numbers of bacteria can encourage growth and survival. And so I'd like you to take a few minutes to do a breakout and think of ideas of why that might be the case. Okay. So let's stop share. So we'll do our normal 5 breakout rooms. So think of anything you can think of to give you the ability to give these microbes the ability to be kind of beneficial or a positive aspect of that. All right, so I'll open those up and let's just take about 3 or 4 minutes to discuss. All right, I'll turn the recording back on. Does somebody want to share? Is there some sort of possible Ali effect you guys came up with in your group?
We talked about that there's safety in numbers, so
Okay.
it's better to have numbers to help fend off possible another competing species. Or
Okay.
then we talked about how large numbers under, like maybe like a catastrophic event, you know, they could help rebuild a lot faster.
Okay, so having a large population might overcome future things. I'm not sure, that's sort of a separate category, but certainly the first item you said in terms of like safety in numbers is, and that's, you know, we see that in the animal world, right? Well, and plants to a degree too, with like a forest, but in the animal world, what do animals do? They tend to group together, right? In herds or flocks, right?
Right.
So we can actually consider microbes to have a similar equivalency. And as you'll see on the next slide, I like to refer to colonies, right, as a microbial herd, right? You've got lots of individuals, all related to each other that are living together. And, you know, if you're a micro major and you've like streaked and restreaked a plate, right? You take your little loop and you stab it into a colony and then you restreak from that. Are you getting every single cell off of that colony to do your streak?
No.
You're not, right? And so that's kind of that safety in numbers piece, right? So some organisms live on even if there's a disturbance. Obviously, a loop is a very artificial disturbance, but you get my point, hopefully. So, okay. I like that. Anybody else come up with some other thoughts on how cooperative interactions might benefit growth or survival?
Yeah, we had one idea, which was kind of relating to what we talked about, like, you know, before the midterm, which was that there's like different species, and then one species could provide a certain thing, and then another species could provide another thing.
Okay. So you could have interactions among species where they're kind of like, are you talking about cross feeding?
Yeah, so let's say one of them produces nitrogen and another one, you know, can't.
Okay. Okay. Yeah, so that wouldn't be an intra-population dynamic, but if you had two different species together, they can, and we're going to talk quite a lot about those kind of mutual symbiotic or mutualistic interactions coming forward. Any other ideas that other groups talked about? Anything within a single species?
In group one, we discussed about symbiotic relationships, like some species may produce something which may help another organism to live. And
Okay.
we spoke about biofilms, which
Okay.
allows anchoring of different species or
Okay. Well, and even within a species, having a biofilm creates a home which can help additional members of that same species. So, yeah, so that's great. Was there anything else that any of the other groups wanted to share? All right, if not, we'll move on back. So, yeah, I think that was good. One thing that wasn't discussed is genetic exchange. Remember, horizontal gene transfer can be very beneficial because it can allow individuals of a species or a subspecies or a clonal line to exchange genes with another. And for this, you need high density, right? You can't have horizontal gene transfer with one cell, right? And so it turns out that being in communities like biofilms likely fosters that exchange across different species. And as we've said, this can even span whole domains, right? Another thing to think of as an example of the Ali effect is something you may have encountered if you've taken bacterial pathogenesis, right? So we know that if you're exposed to one cell of a pathogen, whether it's a virus or a bacterium, fungus, whatever it is it likely you're going to get infected if you encounter one viral particle, for example?
No.
It's not, right? And that's largely because even if it takes hold in your body, your immune system will likely clear it very rapidly, right? And so there's this concept in pathogenesis of a minimum infectious dose, right? So you need to be exposed. And that's actually a big part of why we're wearing masks right now, right? Because if you have a mask, it doesn't cut down every single viral particle. But if it cuts down 80, 90% of them, then you may not pass on COVID to another individual because you're not transferring most of your virion particles, right? So that's kind of an example there. And so from a pathogen standpoint, though, they want large numbers, right? So the cooperative nature of this from a pathogen standpoint is you do better if you have that minimum infectious dose, or you do better if you colonize with more than one individual in a new habitat. Okay. And we already talked about the colony being a microbial herd. All right. Another thing that microbes do, and this was kind of alluded to, I think, in one of the group's answers, was modifying their environment, right? So if you're a species that's growing, some cells may produce enzymes, for example, or exudates, byproducts from their metabolism. And these will be released into the environment, benefiting potentially members of their own species. And then certainly these may crosstalk and benefit others. We've talked about that in the context of microbes, right? In biofilms, like if a phototroph, a primary producer, fixes carbon, that carbon will not only feed that cell, but some of that carbon is going to leak out into the environment and feed heterotrophs. And remember, we talked about this when we looked at that marfish image where you had the filamentous cyanobacterium that had heterotrophs living all over its surface. And those heterotrophs live in a sheath around the cyanobacterium, basically mooching off of the carbon exudates that get released by that cyanobacterium. Similarly, nitrogen exudates or other things like that could be if you release an exoenzyme, right? So probably the classic exoenzyme that micro majors learn about in general micro is the starch test, right? Who remembers what enzyme you're testing for in the starch degradation test?
Amylase.
Right. So amylase is an exoenzyme released by a cell. Let's say a classic organism for this is bacillus, right? They release amylase and it breaks down the starch into glucose, right? And of course, anything can eat glucose. So if one member of the community produces the amylase, everybody in the community is going to benefit from that. Other examples, the production of pH changes through the release of, fatty acids during metabolism, CO2 during metabolism, things like that. Mineral solubilization. We mentioned that biofilm formation provides a home. And so the production of EPS, which remember that's extracellular polymeric substances, the EPS production will allow the organism to, it will allow others to live side by side, right, in this biofilm, okay? UV pigments. So for my dissertation, I studied an ultraviolet radiation blocking pigment in cyanobacteria. And it turns out if you look at a biofilm, it was the organisms that lived on the surface that produced the pigment, because of course they're exposed to the sun's rays coming down first. Other members of the cyanobacterial biofilm living underneath those, they didn't have to make the pigment. And actually that was a benefit to them, right? The pigment made at the top of the biofilm protected everybody. But if you're an organism living underneath, kind of like the umbrella made by the top organisms, you can save energy by not having to produce this pigment. Because of course there's an energetic cost in making a pigment, just like making anything. So this was an example where microbes living together modified their environment and it benefited. So the ones underneath were happy to have the organisms living up above them. If that makes sense. Yeah. So, okay. All right. And so biofilms, oh, this is going to be a little bit wonky because it's not going to go right, is it? This without the animate. Let me see if I can get the animation to work. Okay, can you still see it? Is it still going? Okay.
No.
No, we just see the picture.
Oh no, rats. Okay, yeah, it's doing that thing again. Okay, so, all right. So what I'm gonna do is I will share it. I'll have to kind of, for these next couple of slides, I'm gonna have to be creative. All right, so quorum sensing is a really important concept in microbial ecology. This is a way that microbes talk to each other, okay? Have you guys learned about quorum sensing in any other classes? Yes, no, maybe a little bit, a little uncertain. Okay. So quorum sensing is essentially chemical communications. And so an individual species will produce a little bit of a chemical. And we call these chemicals autoinducers, okay? Within a species, every cell should be able to hear, so to speak, that signal. The autoinducer should induce a response in conspecifics or members of the same species. And if you think about it, each cell produces just a little bit of this, right? So in order for there to be a response, in many cases, you want to wait until you have lots of cells together before you turn something on, right? So what these are population density specific signals. Every cell releases a little bit. And when you get to enough cells releasing a little bit and a little bit and a little bit, then you achieve a quorum. You've probably heard the term quorum before in like a poli-sci class or something like that, right? So it's related to voting. What does it mean to have a quorum? Does anybody know? Have you heard that term before in terms of voting?
Isn't it like you need the top 3 officials? Like if there's 5, you need 3 of the president, the VP, and another voting official.
Okay.
Sure.
Yeah, so basically you need a certain representation, okay? So for example, in faculty meeting for professors, like in the biology department, you know, we make decisions as a department collectively. And you wouldn't want, you know, we have about 30 faculty. You wouldn't want 3 faculty members to make all the decisions and everybody else doesn't get a voice, right? So for us to have a faculty meeting, we actually have to have a minimum of 50% of the professors in the department present to even have a meeting. So that's what we always say at the beginning. Do we have a quorum? And this is true like for Congress, right? You have to have enough congressional representatives there to be able to cast a vote. So in this sense, microbes don't vote, but they release these chemicals, and it's only if they have enough of them present that it'll turn something on. Does that make sense? And it turns out that these, once they were discovered decades ago now, since then we've now discovered that there are dozens and dozens of these kinds of signaling molecules that are produced by maybe not every bacterial species, but most species probably use some sort of signaling like this. Remember those 2-component regulatory systems we talked about weeks ago, right? Where you had the signal coming in from the outside the cell, and then you had the phosphorylation, the kinase activity, which triggered a transcriptional response. These quorum sensing molecules usually function using those. And so these molecules are diffusible, they get released into the environment, and under high density, there's enough of it present to turn on the 2-component regulatory system. And so lots of bacterial functions or microbial functions are under the control of this. An example of that is chemotaxis, right? So one cell migrating for food doesn't make a lot of sense. So they're going to want to swarm or migrate together. And so after there's enough signal, chemotaxis genes get turned on and upregulated in synchrony. That's just an example. All right. So let's see if I can advance that slide. So there's sort of a classic slide here, this picture, right, which is showing bioluminescence. So bioluminescence is one of the most classically studied quorum sensing. In fact, this was really how quorum sensing was discovered. If you're a glowing cell, you don't want to glow if you're only one cell. But if there's millions of cells, then you can turn on the bioluminescence, and that makes sense. And so this picture shows that, okay? All right, let's stop. Why is this? Stop share. Okay. All right. So, and then biofilms themselves are produced via quorum sensing as well. I'm not going to show you a picture of that. You've seen that. Okay. Any questions about quorum sensing?
Yeah, I have a question.
Uh-huh.
Might have missed something, but in terms of that bioluminescence, you said you kind of need like a bunch of cells to be doing that.
Yes.
Is that useful in any way in itself, or is that more like a byproduct of something?
No, it actually is useful. So it's a little bit mysterious, or there's multiple explanations for bioluminescence. There are symbionts that we're going to talk about that are bioluminescents, and they use this as for a variety of things like mate recognition and other purposes as lures, like those deep sea microbes. But of course, microbes evolved bioluminescence before there were animal symbioses, right? So there are species of Vibrio in the ocean. That was what that picture was, that luminesce. And it's thought that it might confuse grazers, right? So there's phytoplankton, algae that are bioluminescent, as well as bacteria that are bioluminescent. And so it's thought that by turning on that light, that it may reduce predation by other groups of organisms. And so that is
Right,
maybe
thanks.
how that evolved. But we don't know exactly because it's sort of hard to prove, right? But good question. But we do know it's under the control of these autoinducers. All right. Now, cooperation is not only found in prokaryotes. And so there's a classic slime mold. So let's see. Looks like this is still sharing. You guys can see this?
Yes.
Can you see the advancing the slide?
Yeah.
Okay. So these are eukaryotic slime molds, right? You've probably heard about Dictyostelium at some point, but they're really cool. So they migrate when food becomes limiting. And it turns out that they use cyclic AMP as a cue. And so they release the cyclic AMP when food becomes limiting. And what that does is it cues the Dictyostelium to form migratory life stages, or amoebae. And what they do is they aggregate together and then they migrate and differentiate. Can you see this picture with the fruiting structures? Or is that, yes?
Yeah.
Okay. So when conditions are bad, you essentially, the Dictyostelium go through this differentiation, forming these migratory amoebae, which can become slugs, which can just glide over to a new location where food might be more plentiful. Alternatively, what they do is they create a fruiting structure. And so these fruiting structures are almost like spore formation in the sense of they create a stalk, and at the top are cells of the Dictyostelium, which then can burst open and release the Dictyostelium cells into the wind so that they can be dispersed to a new location. So essentially, this is two different mechanisms that when you run out of food locally, this microbial eukaryote can get to a new location, either by migrating along the ground or dispersing through the air. And so if you think about it though, only the organisms living up here have a chance of surviving. And so if you're in the stalk, those organisms are sacrificing themselves for the collective good in the formation of that. So it's really kind of fascinating. This is also under two-component regulatory. And it's even more complicated than that. So a paper that came out, I guess about 10 years ago now, showed that they bring their food with them. So it turns out that these Dictyosteliums, they can actually, they prey upon bacteria. And so what they do is when they run out of food, it may be because their bacteria have run out of food, and so they stop growing, and so then the Dictyostelium can't eat the bacteria that they graze upon. So what they do is they take a little bit of the bacteria with them in their slug, and when they find a new suitable habitat, they inoculate the habitat with the bacterium, and then it can grow and feed the Dictyostelium in that new location. And so that was some work that was done, which I find fascinating. Okay, can you see the video?
Yeah.
Okay, awesome. I don't know why it's working again, so we're gonna go with it. So, oops, let me see if this will play. Oh, it won't because I moved it over. All right. Well, there's a video. Maybe I'll try and show it to you guys next time. But there's a video where you can actually see these things differentiating and migrating around. All right, so those are eukaryotes. So Dictyostelium are microbial eukaryotes that form fruiting structures or slugs and migrate. But it turns out that there's a case of convergent evolution, and there are bacteria that do very similar things, although through different independently evolved mechanisms. So myxobacteria are a group of the delta proteobacteria, myxo meaning social or interacting bacteria. And so these are also gliding organisms. They're gram-negative. And they're very unusual for bacteria. You know, with eukaryotes, we see, you know, sort of interesting structures all the time, right? Even among microbial eukaryotes. But in bacteria, these are some of the really interestingly evolved microbes because like Dictyostelium, they form fruiting structures, right? This is not anything like you observe in other bacteria, especially other delta proteobacteria, right? A lot of delta proteobacteria are just unicellular rods and cocci. But this group evolved the ability to form these complex multicellular aggregations. The other thing is these are predators. So Dictyostelium, we said, preyed upon bacteria. And those cells can phagocytize bacterial cells because they're eukaryotic. These guys are also predators, which is weird for bacteria, right? Because bacterial cells cannot engulf other cells, right? So how do you think a myxobacteria might kill and eat another bacterial cell? What do you think they might do? Any thoughts?
Swarm it, release some kind of chemical to lyse it, and then
Exactly.
drink up the juices.
That's pretty much what they do, right? So they release enzymes which allow them to kill other prey bacteria, and then they slurp up the juices. But what do you do if there's no prey bacteria to feed upon? You got to find a new home. And so when conditions are bad, meaning there's not prey around, they form slime trails and they aggregate into mounds using gliding motility, much like what we saw with the Dictyostelium. Now you have to keep these straight for the final. Myxobacteria and Dictyostelium both form fruiting structures. They both glide and move around, but they are not the same. Okay, one's eukaryotic, these guys are prokaryotic. Interestingly, it does not use the cyclic AMP or the same thing we just saw with Dictyostelium. Instead, they don't really chemotax, right? So Dictyostelium was really using chemotaxis, releasing that cyclic AMP as a way of like leading each other together. In this case, it's been well studied, and it turns out that the physicists have determined that it's not a chemical that gets released, it's the way they move in a wave-like pattern that leads them to aggregate. And I don't pretend to understand the physics of how that works, but there's an interesting little paper that I once tried to understand, and at least I could extract that much from it. Once they aggregate, they then differentiate, and that's going to be under a very complex regulation. But they form a fruiting body like you can see here. Sometimes it's a single stalk, and sometimes there's multiple stalks and these fruiting bodies. And in that case, what they do is they release spores, okay, called myxospores. And these, like many spores, are desiccation tolerant, UV resistant, etc. But once again, if you're the organism in the stalk, you're sacrificing yourself because the myxospores are going to be produced up here in the top of the fruiting structure. or they can then be readily dispersed via wind or rain or some other. Okay. So this like sacrificial element is really interesting to think about, because of course bacteria and microbial eukaryotes don't think. They don't choose to do this. So you might ask the question, how does something like this evolve? And so what's been invoked is something called kin selection. So when you're with your close relatives, which of course for the myxobacteria, they're all clonal, they're all identical, right? And so they are brothers and sisters, so to speak. And so it turns out that evolutionary mechanisms that evolve even the sacrifice of certain members of the population, it's okay because your genes are still being passed on. So the myxospores that disperse are identical to your cells if you're in the stalk. And so from an evolutionary perspective, your genes are still being passed on. And so it makes sense. Okay. Now, demonstrating that that's how the evolution of myxobacteria forming these fruiting bodies worked is almost impossible. So, but from an evolutionary theoretical standpoint, it makes sense, right? In fact, with microbes, it makes more sense than it does with some eukaryotic organisms. All right. So let's have a poll question. Okay. Let me see if I can get the polling. All right. So which of the following would be a positive intra-population, within population interaction? Competition, allele effects, quorum sensing, both B and C, or E, all of the above? Which of these would be a positive within population interaction? All right, go ahead and log your answers. Okay, is that everybody? All right, most of you got this one, so that's good. So 93% of you got the correct answer, which is D. All right, exactly. So why is competition not a correct answer? What did we say about competition now repeatedly?
It's negative.
It's negative, exactly, Jocelyn. Okay, so in terms of positive, those allele effects, things like the microbial herd or release of exudates, things like that, those would be positive. Quorum sensing, it relies upon the communications, so those were the best answers. All right, so we've already touched on the fact that there can be interpopulation interactions as well, but everything I've talked to thus far today can be applied to a single, you know, that it can be applied to single species as they grow and interact with each other, cells of their own species. But of course, it's almost never the case that you have just one population. And so what we can look at are negative, neutral, positive, and negative interactions here as well. So neutral interactions are going to be pretty tricky to find, but probably the easiest thing would be resting stages. So if you have spores of a species, other species that are nearby are not really going to care about having the spores. So that would be kind of a neutral interaction. Or if you have a spatial separation, when things are moving through the atmosphere, they're not going to care if they're floating along with something else in the atmosphere because they're not really actively growing, right? We said that was usually just for dispersal. But the vast majority of interpopulation interactions are either going to be positive or negative. And if they're positive, they can be positive for both, which would be a mutualistic, like symbioses, or it can be positive for one and neutral for the other. That's an example called commensalism. And so showing commensalism can be a little bit tricky as well. Usually these are not obligatory interactions. What I like to call the commensalism is usually one organism is mooching off the other. So maybe there's a growth factor or a carbon source that's being released by the growth of an organism, right? One bacterial species is growing, releases a product, and the other species eats that product. Then they benefit, but the first species that was releasing it, they don't really care, right? It's just a waste product for them. It's not a big deal. Similarly, if you're releasing gas like CO2 or sulfide, like a sulfate reducing bacterium releasing sulfide, that sulfide might be used by a sulfur oxidizing bacterium. Now, the sulfate reducer doesn't really care about the sulfide, but the other species benefits from it. Now, that said, you could make the argument that these are mutualistic as well, because the thermodynamics, as you guys probably remember from chemistry, can be altered. If a waste product is removed, it can actually make it easier for the microbe to keep processing and releasing that waste product if it doesn't build up. Similarly, sulfide is a toxic gas. So if sulfide builds up, it can actually inhibit growth, even of the sulfide producing organisms. So in a sense, these truly commensal organism interactions can be really tricky to find, but those are some examples. It's much easier to identify kind of those synergistic or mutualistic interactions. Now, not all of them are obligatory as well. So sometimes they just get together if they're available together, but they don't have to live together. And so this would be a synergistic relationship or a syntrophy. So they feed each other. And so, for example, a methanogen might live with a hydrogen producing fermenter. And the methanogen likes the hydrogen, the fermenter releases the hydrogen, and they cross feed each other. But the methanogen can live on its own and find hydrogen from other sources, and the fermenter can also live on its own. So they're not obligatory mutualists. All right, so we're gone past time already, so I'm going to go ahead and stop there. We've kind of gotten a little behind because of some of the technical difficulties, but we will catch up, I promise. I'm going to stop the recording.